Hi, I think we are ready. We can go ahead and start it. Everyone's in. Good. So good morning, everyone. Welcome to the Clinical Genetics Grand Round. Let me just admit someone. I'm delighted to introduce Zakia Abdelhamed. Dr. Abdelhamed is our first year laboratory genomics and genetics fellow. Since it's the first time to present this format, I'm going to do a kind of briefly introduction, her training and past training and experience. And she received her medical training from Cairo and Egypt. Then she went UK, did her PhD in the neuroscience and medical clinical science in the Leeds University. And after that, she stayed a few years for the postdoc fellowship, and then also came to the state and did a quite extensive research fellowship at Cincinnati Children's Hospital Medical Center. Then she joined Yale LGG Fellowship last August. She published quite extensively for many of these, I call the functional genomics territory modeling the disease in the multiple disease during her training, research training, and the clinical training. So I'm looking forward to her presentation of an interesting, exciting title. Zakia, floor is yours.
Thank you so much for the kind introduction. And it is an interesting title, as you said, and I chose this title because all the years I have been working with mouse models of for brain malformations as it relates to ciliopathy class of disorders. And I always believed in that the face predicts the brain. And it took me like a couple of months in the DNA diagnostic labs working with real people to dispute or to disbelieve in that after 10 years in research, believing in this term. And here is my title, the outlines. So I'm going to talk to you today about the holoprosencephaly human condition and the genotype-phenotype correlation in holoprosencephaly, and also I will touch base on the developmental mechanisms of holoprosencephaly. And this is a case of a 1-year-old patient with holoprosencephaly who have been diagnosed prenatally with lobar holoprosencephaly. And the pedigree here shows that the family was completely healthy. There is no occurrence of genetic conditions, as you see, except for the prostate cancer, the early diagnosis of prostate cancer in the grandparent. And the patient was 1 day old. She was a twin, chorionic diamniotic twin with her brother. Her brother was completely normal. And the mom denies any history of serious infection or diabetes mellitus during pregnancy. And the birth was uneventful. The Apgar score at the time of birth, 1 minute and 5 minutes, was perfect. And the birth weight was okay. Everything was perfect, and the consanguinity was denied apart from the perinatal diagnosis of the lobar holoprosencephaly. And the postnatal MRI have confirmed the perinatal diagnosis, showing the patient here. This is a patient scan showing fusion of the gray matter at the frontal lobes and absence of the septum pellucidum, the structure in the midline here that you see in the control. And the patient lost this. And also there is evidence of corpus callosum dysgenesis or agenesis. On the dorsal side of her brain, she showed also incomplete interhemispheric fissure and absence of the phalanx cerebri, which is the part of the meninges at the dorsal side. And at the level of the craniofacial assessment, she was kind of very, very normal. There is no evidence of no facial clefting, and the only questionable hypothyroidisms that was showed by MRI, but not on physical exam. And she was also have a deep philtrum on her face. So, and when we come to the endocrinology and the pituitary assessment, the anatomy of the pituitary was also unremarkable. The pituitary gland lies here on the floor of the forebrain or the roof of the nasal cavity, and it seems okay for age and sex. The infundibulum of the pituitary gland also looks normal. And when the endocrine assessment shows that she had adrenal insufficiency as evidenced by low cortisol levels. However, she also showed normal response to adrenocorticotropic hormone stimulation, and this indicates that she don't have a primary adrenal problem, but it indicate dysfunctional hypothalamo-pituitary axis. Growth hormone was high at the time of birth, but later assessment it normalized. All other pituitary hormones were normal for age and sex. And as you know, that once a congenital anomaly is there, the fetus or the baby should be investigated for all multi-team management. The investigation of the visceral organs and the heart found a patent foramen ovale in her heart. And the neurology exam was unremarkable. Surprisingly, there was no microcephaly or seizure. And what's holoprosencephaly? Holoprosencephaly is a devastating forebrain abnormality caused by loss of midline tissue. Midline either in the dorsal side or the ventral side of the forebrain. This is the holoprosencephaly. So we don't have, the patient normally fail to develop the dorsal midline or the ventral midline or both. And is actually a very common condition. It affects 1 in 250 embryo at the time of pregnancy. If studies have done had found that this is the incidence, but when it comes to live-born, it is 1 in 10,000 live-born. And because the most severe cases usually lead to spontaneous abortion, makes the incidence less common in the live-born. And according to the clinical classification, it can be syndromic or as a part of the syndrome, or isolated, usually they call it above the neck. And or according to the anatomical classification, as I mentioned, it can be ventral, and this is also called the classic holoprosencephaly, and I will go into much more details about that soon. And if it affects only the dorsal side of the forebrain, it is called the non-classic, and it is called also middle interhemispheric variant. So the defect, as I mentioned, it is due to during embryonic development. It occurs from the 3rd to the 5th week of gestation, and this happens when the prosencephalon or the forebrain primary vesicle fail to divide into right and left cerebral hemispheres or telencephalon hemispheres due to lack of development of the interhemispheric fissure. And this, as you see, it occurs very early in development from the 3rd to 5th week. So the diagnosis usually very early. So according to the degree of lack of development of the interhemispheric fissure or the dorsal and ventral midline, in cases of the classic holoprosencephaly, we can see it divided into 4 classes. That extend like a continuum of severity from a lobar to a microform. So you could see a patient with incomplete absence of the interhemispheric fissures and complete fusion of the right and left cerebral hemisphere on the dorsal and ventral end. All the forebrain is one vesicle, and the lateral ventricle, right and left side, is now monoventricle. As you see here, one single cavity. And next to that, there is the semilobar holoprosencephaly, where the separation occur normally at the posterior end of the forebrain, but the ventral and parietal cortex and also the ventral structure also lack any kind of separation. This is the most common type of all three cases, all three conditions, the semilobar. And here is the lobar holoprosencephaly. The second in severity to the semilobar is the lobar. The separation of the posterior occur normally, and also the separation of the frontal cortex occur normally, apart from the ventral side, where you see fusion of the gray matter at the frontal end. And the two lateral ventricles separate to some extent. And the last thing is the normal brain anatomy. So, but how we can call this holoprosencephaly, because they have other evidence in the facial structure, like the single central incisor or midline philtrum. And when I come to describe the craniofacial abnormalities, there is always correlation between these forebrain anomalies as opposed to in correlation with the facial malformations. So you see here cyclopia and the proboscis and the totally malformed face in this allobar holoprosencephaly babies all the time. And when it comes to the semilobar, the slightly milder form, we see milder presentation and along the facial structure with hypothyroidisms, nasal bone dysplasia or aplasia, and midline cleft in the palate as well as in the lip. The lobar, usually they have again milder faces, but you still see some hypothyroidisms, and probably also can have facial cleft. For the microform, this is the form that you only have facial malformations, but very mild, very mild, but no brain malformation. So, and this is a continuum of the classic holoprosencephaly. The non-classic or the middle interhemispheric variant, the separation of the basal forebrain or the ventral forebrain occur normally, but the anterior frontal lobes and the occipital regions lack the separation, and sorry, the posterior frontal and the parietal region fail to separate, but the frontal and the occipital lobes, they separate normally. And the rarest form of it is the septo-preoptic dysplasia, and where no separation is restricted to the septal and the preoptic area on the ventral side of the forebrain. Additional neuroanatomical malformations that can frequently develop in those patients is the undivided salami, agenesis or the genesis of the corpus callosum, absent septum pellucidum, absent or hypoplastic olfactory tracts, and also absent or hypoplastic optic nerves and tracts. Macrocephaly sometimes occur if the patient also develop hydrocephalus. Microcephaly is the more common. Neural tube defect sometimes occur. Dandy-Walker malformation occur, especially if the gene is involved in the hindbrain development from the beginning, as I will touch on that later on. Neuronal migration anomalies is also very common, and abnormal circle of wheels also common. And on the clinical side, these patients usually develop neurocognitive impairment. The top row is the most common complication, seizures, and also diabetes insipidus due to defective posterior pituitary and antidiuretic hormone secretion from there. And also the cleft lip lead to recurrent infection due to aspiration pneumonia. Cleft lip and the palate leads to recurrent infection, aspiration pneumonia, and also feeding intolerance or feeding difficulties. And they also commonly develop other congenital malformation, cardiac or renal, and others. At the genetic side, so the genetics is highly heterogeneous, and so there are extreme locus and genetic heterogeneity in this condition. So chromosomal abnormality accounts for about 50% of the cases, mostly trisomy 13. That causes about 75% of the holoprosencephaly due to any chromosomal abnormality. Trisomy 13 is the most common cause of that. Trisomy 18 and triploid is also holoprosencephaly can be as phenotype there, but is rare. Next to that is a submicroscopic chromosomal abnormalities, the pathogenic copy number variants that accounts for 10%, about 10%. And I will go through this very soon. There is also 25% of the cases are due to monogenic syndromic condition like Smith-Lamilli and Mickel-Guber syndrome. And about 10% is or 12% due to environmental causes. And what is more interesting to us today is the monogenic causes of holoprosencephaly. So there are 4 genes that are most commonly causing holoprosencephaly or most commonly mutated in patients with holoprosencephaly: sonic hedgehog, ZIC2, 63, and TGIF1. And there are other genes that has been discovered, and I will go through all of them today. But they are rarely causing or rarely detected as mutated, but they are there. Regarding the pathogenic copy number variants, they are non-specific and detected in all chromosomes, as you see here on this karyotype, hypothetical karyotype that shows the anomaly, the pathogenic copy number variants. But there is some that are highly recurrent, and these are listed here. And in most of them, there is a candidate gene have been discovered in the region. Like for 13q deletion, we have ZIC2, and we have on the chromosome 7 deletion, we have sonic hedgehog have been discovered as a candidate gene in the region. This slide lists the monogenic syndromic condition in which holoprosencephaly can be a phenotype, and this I divided them according to the mode of inheritance. They can be autosomal dominant, as in these. All of these syndromes are autosomal dominant, and holoprosencephaly is a phenotype in all of them. Autosomal recessive, the most common is the Smith-Lamilli-Obitz syndrome and the Mickel-Guber syndrome. And caudal dysgenesis, it's also holoprosencephaly have been reported, but the mode of inheritance is still controversial. And I will come to the more interesting part, the monogenic causes, the most common. These are, as I mentioned, the most common gene causing holoprosencephaly. And according to the craniofacial, the incidence of the sonic hedgehog is the most common cause, followed by ZIC2, and an order of how they develop in human. Sorry, how the mutation is being, how often is the mutation is being discovered in human patient. Craniofacial malformation, they can cause across the spectrum from cyclopia and proboscis and totally malformed faces to microform or middle central incisor. And all 3 of these that I did not highlight, except for ZIC2, which can cause very, very mild phenotype and nothing like the ones on the top of the spectrum. Other associated anomalies, in addition to the spectrum of holoprosencephaly, the sonic hedgehog is commonly causing renal abnormalities, and also ZIC2, same thing, and interfamilial variability is highly common in 63. And this slide showing the rare forms, the rare genes that have been involved. In total, we have like 17 genes causing holoprosencephaly. 4 that I just described is the most common, and then these are also involved, all of them causing across the spectrum of craniofacial malformation, they causing across the spectrum of holoprosencephaly, neuroanatomical malformation. And this one is also can cause diabetes and pancreatic exocrine dysfunction and ossicle and sensorineural and conductive hearing loss. And some of these, like the CDON1, it's found like downstream of sonic hedgehog signaling. That's why it is involved in the condition. However, it is also rarely causing craniofacial malformation along with the DLL3 and still gene, and this one is inherited as an autosomal recessive. Here I show the genotype phenotype correlation in holoprosencephaly. So I focused on the four most important or the major gene causing the condition. So ZIC2 is the most commonly causing structural brain malformation, while the microform is occur in sonic hedgehog as equivalent to the nearly equivalent to the when it causes structural brain malformation. Of the structural brain malformation, the most common in ZIC2 is the semilobar holoprosencephaly. And also in ZIC2, we have the middle interhemispheric variant. It seems like the ZIC2 is the most common cause of middle interhemispheric variant in those patients that develop the holoprosencephaly. So testing for ZIC2, if the patient have middle interhemispheric variant, we would suspect that ZIC2 is the mutant gene based on these findings. And usually there is a correlation exists between the facial finding and the degree of the brain anomalies and the survival, except for these four genes. So this is the four genes in which the face does not predict the brain. And the mode of inheritance, generally, it is in this showed here in this hypothetical pedigree that is autosomal dominant with incomplete penetrance. So you can see that affected parent is coming, affected patient coming from an affected parent, and also some of the pedigree can, some patient can be highly affected and pass away, while others are less affected. This is variable expressivity, intervariability, interfamilial variability is very common, and as you have seen, locus and genetic heterogeneity is very, very extreme. Environmental factor leading to or that have been linked to development of holoprosencephaly include maternal diabetes, accounts for increase the incidence to like 200-fold. So it's very, very teratogenic, the maternal diabetes for the patient, or gestational diabetes if the mother have gestational or pre-gestational diabetes. Holoprosencephaly is highly expected to be in the pregnancy. And also maternal infection. Also, there is the studies that link the holoprosencephaly conditions into assisted reproductive technologies. Twinning and the female sex is also there. And as a reminder, our patient was a twin and she was the female girl. And thyroid hormone use during a pregnancy and the pesticide exposure. Retinoic acid, statins use, and the maternal hypocholesterolemia are also reported, alcohol use and prenatal exposure. The first three is because statins and the cholesterol is needed for sonic hedgehog post-translational modifications. So if the patient have hypocholesterolemia, or if she's in use of statins, this will reduce the cholesterol level in the blood, and also leads to defective post-translational modification of sonic hedgehog that is needed for its activity, and so that the phenotype could be explained on the basis of that. And the retinoic acid exposure, because it opposes also the sonic hedgehog activity. Life expectancy of those patients, according to or contrary to the popular belief that all of these patients die very early in their life, this is actually true for the lobar holoprosencephaly when there is severe craniofacial anomalies. These patients are always passed away, but the rest of them, the incidence of the semilobar holoprosencephaly in children, adolescent, and adults is about 50% in across the lifespan. So it seems like this patient lives with, of course, with the assistance of or the management of the medical teams to correct the facial anomaly, like in this patient in D, and also by full dependence on caregivers is reported in 60% of them, but they live. So how we diagnose these patients? Perinatally, of course, the detailed radiological examination once suspected, ultrasound or the high resolution 3D ultrasound and MRI can be needed for the milder forms to confirm the anomaly. And once confirmed, the patient is advised to do either the invasive or non-invasive testing, genetic testing. The non-invasive is highest sensitivity and specificity for chromosomal abnormalities, but bear in mind, this is still a screening method, not a test. Not a diagnostic test, and the chorionic villus sampling and amniocentesis to perform karyotype analysis or chromosomal microarray, SNP array. And if all of this didn't show any abnormalities, we prefer to do whole exome now because there are so many genes and testing all of these will be very troublesome. And so if all the karyotype came back normal and the chromosomal microarray is normal, so exome or genome can be the choice to go. Going back to the case, what happened to her? She had non-invasive perinatal testing and it was negative. And the parent waived the invasive testing because they don't wanna, it wouldn't change their mind if she have, even if she have any problems. So they wanted to have her. That's very kind. And the whole exome sequencing was ordered once we saw her. Dr. Bell have ordered the whole exome and we identified a frame shift variant in ZIC2 gene. And this, as a result, the whole exome sequencing detected the 10 base pair deletion novel variant. This is not reported before in literature. It is a 10 base pair deletion, as you see here, that causes a frame shift. And this deletion is a frame shift. This deletion causes a frame shift, and the frame shift is the most common type of mutation in ZIC2. So the missense are here shown in red, and this black squares are the frame shift, and this is the most common compared to all other types of variants. And our mutation exactly was affecting, so I was very happy to see the frame shift. I saw it's pathogenic, but it came up to be in the third exon, affecting a mutational hotspot. It is definitely likely pathogenic, but it is not like in the first exon or second exon, but it affected the alanine repeats. Any changes in the alanine repeat, there are 15 alanine residue, amino acid residue in this in the C terminus of this protein, and it deleted 5 of them, and it causes a frame shift in of the rest of the protein, and additional 20 amino acids were added before the introduction of the stop codon. So the variant causes protein length changing and abolished the normal stop codon and affected the highly conserved alanine repeat. They are 15, need to be 15, not more, not less. So we deleted 5. This variant deleted 5 of them. And before, there is multiple reports that any change in the alanine repeats are pathogenic. And when we come to the ACMG classification, the variant was absent from the population database. As you've seen, it causes protein length changing and it affects a mutational hotspot, and this qualifies it to be a likely pathogenic variant. And in ZIC2, and ZIC2 is causing a type 5 of holoprosencephaly, and here is just pictures of the patient, of the particular patient of ZIC2. It can cause the aluber, the semilubar, and the lubar holoprosencephaly. In addition to, they most commonly have some type of large dorsal cyst or arachnoid cyst at the posterior fossa of their brain, very large. This is a very characteristic occurrence in ZIC2 patient, holoprosencephaly patient. And as I mentioned before, it is a major cause of the middle interhemispheric variant, and we will know why later on. And 70% of those patients of these variants are de novo, and because they lack the craniofacial, as I will come into it very soon, they are more likely to live. And the germ-like mosaicism is not very rare, and genetic counseling for all patients is always recommended. And here I am showing the craniofacial malformations that you might detect in patient with ZIC2 in particular. So they never have the proboscis or the cyclopia, but they can have the bitemporal narrowing, even if they have the alubar phenotype. So this kid on A showing just bitemporal narrowing, and I believe that's it. But she had on her brain alubar holoprosencephaly, complete absence of the midline structure, and one single ventricle in the forebrain. And they have could have the hypotolorisms in eye here, as you see, and they also they could have larger ears, very non-specific and very mild facial phenotypes. And the one that is very commonly as well is the upslanting palpebral fissure. This is always seen there in most of them. And so comparing the phenotypes of the ZIC2 mutant, and we think that this is the best fit or the best phenotype match to our patient. Our patient have holoprosencephaly and very mild cranial facial abnormalities. So this is best to explain our patient phenotype, and that's why we reported the variant. So I will, I just showed you the holoprosencephaly condition and the genotype-phenotype correlation, and then I will touch base on the developmental mechanisms of holoprosencephaly. So as regards to ZIC2, ZIC2 is a member of 5 genes that is a transcription family of transcription factors that is zinc finger proteins of the cerebellum. This is their name, and they occur impaired along the chromosome, along a couple of chromosomes, apart from ZIC3, which is occur in isolation on the X chromosome. ZIC1 and 4 occur on the chromosome 3, ZIC2 and 5 on chromosome 13. And an interesting observation that they are transcribed in opposite direction, as you see with the direction of the arrows here. And it is known that they also have very overlapping expressions during development, but very specific, as evidenced by showing that some of them, not all of them, cause the same phenotype. If their overlapping expression is definitive, so they would cause the same phenotype, all of them. But you see that in ZIC2, ZIC1 and ZIC4, they cause Dandy-Walker malformation. ZIC2 causes the holoprosencephaly, as I just showed it. And ZIC3 is the most common cause of heterotaxy and the congenital anomalies in human. So the expression of them is overlapping, but distinct at the same time, is evidenced by different human phenotypes that happen when any of these genes are mutated. ZIC5 have not been linked to any human condition so far. And here I am showing the ZIC2 messenger RNA expression during development. It is expressed during gastrulation and also during organ formation and during neuroectoderm initiation and primitive streak. And here during organogenesis, you see it in the dorsal ectoderm and also in the lateral mesoderm and in the somatic mesoderm. Later in development in the brain, you still see it in the ventral forebrain and the dorsal forebrain, and it is known to cause to be repellent for the developing axons from the middle line. Very interesting observation that has been observed in mice. So how the dorsal and ventral midline develops. During neurulation and formation of the neural tube, we know that the neural plate have to form a neural groove and then fuses to form the closed neural tube. Here I'm talking about the caudal neural tube. So, and then according to, under the influence of this complex signaling system from the dorsal side, we have the BMP signaling, Wnt signaling, ZIC and GLI3 at the dorsal side, and the ventral side we have sonic hedgehog that is secreted from the notochord, and then this secretion initiate the secretion of sonic hedgehog from the ventral from the floor plate or the ventral neural tube. And the sonic hedgehog, as you know, is a typical morphogene. It is secreted from a source and then diffuses to pattern the surrounding tissue in a concentration-dependent manner. And so interaction between the dorsal marker and the ventral sonic hedgehog, this leads to the formation of the dorsoventral midline. And the final process is the formation of the floor plate and roof plate. So this is the midline dorsal. This is corresponding to the dorsal midline, and this is the ventral midline. What actually happened under this complex signaling system, the midline cells undergo high level of cell death and reduced proliferation, and this leads to benching of the from the forebrain vesicle or from the neural tube, developing neural tube, and this leads to the formation of the floor plate and roof plate corresponding to the dorsal and ventral midline. This system continue to the forebrain. This is caudal neural tube, and this is the most cranial neural tube. The same paradigm continues. We have the sonic hedgehog in the procaudal plate here, and this is the most cranial end of the notochord, and we have fibroblast growth factor at the rostral end of the developing forebrain. This is the only difference between the caudal here and the cranial neural tube. And here we have at the back the PMB and ZIC and Wnt, similar to what we have in the caudal neural tube. So we need to form the ventral midline, dorsal midline, to be able to divide the forebrain into right and left cerebral hemispheres. So what happened in the ZIC2 mutant? It is, as I just showed you, it is expressed in the dorsal with the Wnt and the BMB. Here, what happened? It leads to, when ZIC2 is not there or there is mutation affecting the ZIC2 gene, the high level cell death and is not happening in the dorsal midline. And this is needed to happen for the mesodermal cells or the migrating neural crest cells that coming from the hindbrain to fill the gap here, and later on will form the meninges and the phalanx cerebri. and also the head mesenchyme, everything will be deficient if ZIC2 is mutant, and if this step didn't happen from the beginning, the cell cycle arrest and reduce the proliferation. And it's believed that ZIC2 do this by opposing the action of the canonical Wnt beta-catenin signaling in here by interacting with the TCF factor, and this leads to inhibition of the gene expression downstream of the Wnt beta-catenin signaling system, which is responsible for the mitogenesis and advancing the cell cycle. So if this didn't happen, if ZIC2 is absent, you leave this Wnt unopposed, and we lose completely the dorsal midline development. And the dorsal midline development leads to choroid plexus development and the cortical hemisphere, and this rarely develop in the mouse model of holoprosencephaly of ZIC2 mutant causing middle interhemispheric variant holoprosencephaly. This is diagram coming from or depicting, summarizing the finding in the mouse of ZIC2. When we come to the ventral midline, it is under control of the brochordal plate, and in combination with both things happen together, the eye field and the septation or separation of the ventral midline of the developing forebrain happen together simultaneously under control of sonic hedgehog shown here in red. From this white structure that is called the notochord. So if you have normal development, the eye field will divide into two eyes, and the forebrain, overlying forebrain will also develop into to right and left cerebral hemispheres. But if we have reduced the sonic hedgehog signaling, we may have the forebrain dividing, but the eyes will show hypotelerisms. And if there is no sonic hedgehog completely, we will have the cyclopia. And if there is no brochordal plate, if there is defective brochordal plate development, these patients will also develop cyclopia and holoprosencephaly. So the clinical manifestation of defective process of brochordal plate arrest or sonic hedgehog loss of function or overactivity would be cyclopia and the hypotelerisms, hyper or hypo. If you have so much sonic hedgehog, we have hypertelorisms. If we have little sonic hedgehog, we have hypotelerisms. And this is also usually accompanied by facial clifting and holoprosencephaly spectrum of malformation. With regard to ZIC2, this was very controversial, how ZIC2 can cause the structural ventral forebrain division defective. Is mainly expressed at the dorsal side, but it is found that it is under control. This is the most accepted or the most easy to understand mechanism of ZIC2 causing ventral structural brain malformation, is that it is under control of sonic hedgehog and the ventral midline complexing with, because it is very, very similar. The zinc finger protein on ZIC2 is very similar to the GLI members of transcription factors that is mediate the sonic hedgehog signaling. And once it is combined with the GLI, it is knowing that it can translocate the GLIs into the nucleus. And once it is in the nucleus, it activates FOXA1, and the FOXA1 causes inhibit proliferation in the ventral midline or the floor plate cells, and this leads to development of the midline structure. If we don't have ZIC2, we lose the FOXA1 and we lose the development of this. Also, another paper have showed that it is under control of the nodal signaling, where it complexes with the SMAD2/3, and also leads the end product of that, it is leading to FOXA2 initiation in the floor plate and the inhibition of proliferation to form the ventral midline. And this I am just, as this slide is showing to how controversial in literature the role of ZIC2 in the ventral forebrain development. So a group in 2001 showed it is in the brocaudal plate downstream of sonic. That's it. And eight years later, another group showed that no, it is in the ventral, it is much earlier than the brocaudal plate stage of development. It is in the primitive streak, in the primitive node or the primitive pit, where in ZIC2 mutant, they found that there is transient defect in the primitive node that leads to arrest of the brocaudal plate. And this is that leads to the holoprosencephaly phenotype. Later on, this also other group have showed that it is much even earlier in the primitive streak stage, where all the cells of the brocaudal plate is initiated. They initiate here, migrate all the way through until it reaches the most cranial end of the embryo that below the developing forebrain. So these group have showed it is even earlier before the primitive node and before the brocaudal plate development. It is here in the primitive streak. But all of these are hypotheses in it's done in mouse. So how relevant is this to the human patient? We still don't very sure. Analysis of the human mutations indicated that the dorsal midline cells are more sensitive to loss of ZIC2 because the milder mutations, the deletion, duplication, the in-frame deletion, duplication, and the intronic variant can cause this middle interhemispheric variant. It's a very mild form, but you don't, when it's frame shift, you don't see any of the other subcategory of holoprosencephaly in these. So it seems like these dorsal midline cells, very milder loss of function leads to development of the middle interhemispheric variant. But to develop the ventral or classic holoprosencephaly in those patients, you need a very strong mutation that affects the protein structure, or even mesence is not reported in the middle interhemispheric variant. And this slide shows the other associated clinical conditions that is, or phenotypes that accompany or occur, most frequently occur in ZIC2 mutant patient. And you see they have hydrocephalus, they have neural tube defect and skeletal anomalies and renal anomalies, and 5% of them, they have complex congenital renal and skeletal in addition to the holoprosencephaly. However, it is always described that ZIC2 is a cause of isolated holoprosencephaly. So, and there is no syndrome describing this so far or to date. And all of these comorbidities also recapitulated in mouse models, and I think it is there. So many people also showed that it is under control of multiple essential embryogenesis, essential signaling system, and that's why it can cause multi-system involvement. And some other people think that it is also downstream of the cilia, and especially the cilia in the node. So this is the primitive node, and this is a primitive streak of the very early stage developing embryo. And if you look at the node at higher power, you see that they have this nodal cilia. Nodal cilia is a subtype of the motile cilia that lacks the central pair of microtubule doublets, as you see in cross-section here. And this nodal cilia is very important. They found recently, like 20 years back, just recently, I mean, that this cilia is very important to, they beat, in spite of the like the central pair, they beat, but the rotatory movement to direct the fluid around the fetus into the left side, and this is detected here. This is nodal signaling, and if you have the cilia beating here, this will direct it. It moves from right to left, so it directs the fluid containing the morphogene to the left side, and this leads to the breaking of the organ symmetry during early development. And breaking of the organ symmetry is very important embryonic step for multi-organs, including the heart. If the heart looping is not directed to the left side, or is not directed to the left side right during early heart development, this will be accompanied by a plethora of congenital heart malformation. But they found that if heterotaxy, we have the congenital heart malformation. If situs inversus two tails, we don't have any heart malformation, but this is beyond the scope, so I cannot go through all of this. But the function of ZIG2 here in the nodal cilia is to have been also described in mice, that it is also important for the nodal cilia to be beating and distributing all of these left side markers to the left side of the developing embryo, and that's why the mouse develop also congenital heart. So there is reported that it could be downstream of the cilia, but again, this is still under investigation and under controversy. I think it is because for brain defects is a very common phenotype in ciliopathy conditions, as well as the congenital heart in the multi-system involvement. But again, as I mentioned, this is still in the process of research. So this is the summary. I think you may agree with me that the most important stage and the person life cycle is not the birth or the marriage or this, but the gastrulation. It's the most important step. If this happens correctly, we will be fine. If not, will not. And ZIG2 loss of function variant are the second most common cause of isolated holoprosencephaly. And the dorsal midline cells are more sensitive to ZIG2 partial loss of function. And a severe loss of function is a mechanism of disease in classic holoprosencephaly due to ZIG2 variant. And then clinical exam, of course, provided in this patient, provided robust and time and cost-efficient genetic diagnosis for our patient, and should be applied for all patient, holoprosencephaly patient who don't have chromosomal abnormality. And now I can say that the face predicts the brain right, but not always the case. There are some genes that are not, where you see brain malformation, but not face anomalies. And here is my acknowledgement slides. I would like to thank all the faculties involved in my training, Dr. Bale, Dr. Zhang, and DQ, and everyone here who have contributed to my training and or will contribute to my training. Also, all members of the DNA diagnostic lab, they helped me a lot all the time and every time. And thank you all, and I am happy to take any question.
Well, that's a great, very comprehensive review, Zakia. Let's open for question or comments.
Yeah, I have one comment and one question. My comment is that among animal models for holoprosencephaly, the most important one is the sheep model that has to do with eating corn lilies. You should look it up. That's the most important animal model. The other I was going to ask you about is, like, your last point was exome sequencing was a cost-effective, quick, efficient way to diagnose holoprosencephaly. Why do chromosomes ever at all, chromosomes or CMA, when you're dealing with this diagnosis? what does that add that the exome doesn't give you?
I'm sorry, what the chromosomal?
Oh, your second to last point, clinical exome provided robust, time and cost-efficient genetic diagnosis to HPE patients. But you said that it should be used in patients who don't have a cytogenetic abnormality. Why not just do the exome? Why do you need to do cytogenetics first?
Because of the copy number variant that
we find copy number. We can see
we can find
yeah. And virtually every copy number variant.
Yeah, that is right. Yes, absolutely. Yes, I agree with the sentence. I agree that the copy number variant can be detected by exome, but this is the trend now is to send the cells or the CVS amniocentesis cells to karyotyping and then microarray, and then if nothing happened, we send the exome. But well, I totally agree that the exome could solve the patient, could give answer to the patient about the copy number variant and the chromosomal abnormality as well. Yeah.
Yeah, I'm challenging that model of care. That's all. I mean, I don't agree with it anymore.
Okay.
That's a great point, Dr. But hoping that people are not familiar with us testing technology, kind of moving forward, kind of realize that's the sort of future. Other comments, question?
This is Miriam. I don't know if you can hear me, but
yeah, we can hear you.
Yes.
I just had a response to what Alan said, that if it's a prenatal sample and you can do FISH and very quickly establish that this is trisomy 13, it might help with a quick decision about pregnancy termination.
Yeah, I mean, I agree. It's all a matter of speed. You know, it has, I mean, the effectiveness of the diagnostic measures are all pretty much the same. It all has to do with speed. And so that's I mean, it's just a question. I mean, if we, certainly in the postnatal setting, usually you don't need that kind of speed. In the prenatal setting, you know, there's an argument for it.
I think practically, sort of logistically also right now, I think that many insurance, I guess, the also comes to the insurance coverage for sure. Need a lot more education, sort of want to only cover the microwave, but not cover exome for sure. That created some challenge in the real world. So, I was kind of question. Sounds like for the holoprosencephaly, you do have some kind of early acquired maternal exposure. So has anyone kind of look at whether for this mild form and low form actually it's a somatic mutation origin?
I'm sorry?
Is has anyone kind of investigated for this mild and low form actually it's a somatic mutation origin, not germline mutation? Because I mean, you can have acquired sort of early exposure environment factor across this similar presentation. So it sounds like that's a later event could also be the cause, but such as somatic mutation. So
right, yeah, because it's confined to the forebrain. Yeah, and very limited. Yeah, it's possible, but as we don't know. We don't know.
No one has looked at like a publish any small scale study. Okay.
No. There's somatic variant is in holoprosencephaly. There is no report of. Yeah,
probably less common than other like a mesencephaly, hemimesencephaly. That's more common in somatic mutation.
Yeah,
and this definitely is much early event for sure. Any other question or comments? Well, I have one more question for the overall. What's your? I think you have a bar graph and shows the trisomy definitely is a common one, and the ZINC2 is definitely on the trisomy, on the chromosome 13, that which is deletion. Is anyone suggesting ZINC2 is a very dosage sensitive gene, and also is a key contributor for the trisomy 13 contributor this phenotype, or as no one knows? No one invested in that.
Overexpression or
ZINC2, yeah, it's overexpression ZINC2 in any animal model also cause similar phenotype, or no one have ever?
No, overexpression causes cancers.
Cause cancers. Not
developmental. Yeah, it seems like the trisomy 13. You think that the overdose of ZIC2 can be also leading to the holoprosencephaly, as well as the deletion, but no, this is excluded.
Great, we are over 2 minutes. Thank you so much, Zakia, for the comprehensive review, and hope you all enjoy that topic, and we'll see you next week.
Thank you. Thank you.